This lecture will cover cell membranes. So, be able to define what is a cell, what is a plasma membrane, what is a phospholipid, what are integral and peripheral proteins, what is diffusion, facilitated transport, and active transport. Compare endocytosis, exocytosis, and know what a receptor is. Be able to compare the different types of movement across the cell membrane and diagram how a sodium-potassium pump works. We're going to be starting a unit on cells and their components with this lecture. So first, we're talking about eukaryotic cells. Now, eukaryotic cells are what make up all humans and other mammals. Eukaryotic cells have a cell membrane, a nucleus, a cytoplasm, and organelles. This is in contrast to prokaryotic cells, which make up bacteria. Prokaryotic cells do not have a nucleus or organelles, and we will be spending the next lecture looking at nucleus and organelles. All living things are made of cells, but not all living things are made of eukaryotic cells. As I mentioned, bacteria look very different from the cells we have. So why do we have many cells and not just one large one? Well, this comes down to the issue of surface area to volume ratios. With one large cell, there is less surface area compared to the volume than 8 small cells, which will have more surface area. Since all exchange occurs across surface area, this is important for exchange rates. Also, it takes longer to diffuse to the center of a large cell than it takes to diffuse to the center of a small cell. And this diffusion, in coordination with the circulatory system, is going to mean that every cell will have a relatively small distance between it and its center and whatever is supplying it. And I mentioned the circulatory system because there is differentiation between different types of cells with different functions. So we have here muscle cells. Muscle cells have a lot of cytoskeleton, and muscle cells can contract. Nerve cells have extensions on them and things called axons and dendrites, which allow them to transmit electrical impulses. We also have cells lining the tubule of a kidney, endothelial cells, as we'll cover later, and these allow the exchange of things in one direction. So these different functions of cells are one reason it is important to have many different cells. And for some of these, I've been talking about what occurs on their membranes. And membranes separate the interior of a cell from the exterior of a cell. Membranes are made of a lipid bilayer, which we'll talk about in a moment, that is made of amphipathic molecules, meaning having a polar head and nonpolar tails. These are phospholipids, which we'll get to in a moment. There are also cholesterols in the plasma membrane that modulate the fluidity of the plasma membrane to make it more or less fluid at body temperature. Proteins are embedded in the membrane. So these proteins have various functions, such as signaling to other cells, receiving signals from other cells, or allowing things through the plasma membrane. Phospholipids are what make up this lipid bilayer. As in a previous lecture, we mentioned that lipids are nonpolar, and these carbon and hydrogen containing molecules are nonpolar fats. So these fatty acids make for a nonpolar side that does not interact well with water. But the phosphate group that is attached to the glycerol on the head of these makes for a polar side that interacts well with water. So you have the tails stuck together in a nonpolar area, and the heads facing the water inside the cell and the waters outside the cell. So this phospholipid bilayer and the integral proteins that are put in it, more on that in a second, make it a selectively permeable plasma membrane, meaning some things are allowed in and some things are not. So this is what the plasma membrane looks like. We call it the fluid mosaic model because the membrane is a fluid of phospholipids. Not a liquid like water, but a fluid in which things can move around a bit. And it is a mosaic of different components. Different sides of the cell will express different proteins in them, giving cells definite sides. There are integral proteins, which have parts on the inside of the membrane and on the outside of the membrane. They go through the membrane. Peripheral proteins are located only on one side of the membrane. We also have glycoproteins. Now, integral and peripheral proteins can each themselves be glycoproteins. This is a term used for any protein that is attached to a carbohydrate group. These are such things as the ABO blood groups, which are carbohydrate signals on the end of a glycoprotein. Let's look a little more at integral proteins. Integral proteins have polar and nonpolar segments. What this means is the nonpolar segments can be interacting with the lipid tails of the phospholipids, and the polar segments can be interacting with the polar heads of the phospholipids and the waters inside and outside of the cells. Now, what makes them polar or nonpolar? Well, that's just what amino acid groups are present. So nonpolar amino acids like isoleucine and valine will be facing those nonpolar lipid tails inside the lipid bilayer. You look at those little barrels right there, and those are what's called beta barrels. So beta barrels are beta-pleated sheets that are arranged like staves in a barrel, allowing a hole in the middle through which things can pass. We'll get back to that in a few. And these are what are called transmembrane domains. They're domains that go through the plasma membrane. If it does not go through the plasma membrane but stays on one side, we have peripheral proteins. These are anchored into the membrane, but only on one side of the membrane. Blood type factors are glycoproteins that are on the outside of a cell. On the inside of the cell, the cytoskeleton, which we'll discuss in a future lecture, may actually be connected to the membrane by peripheral proteins that are anchored into the membrane. So how do things get across this membrane? Well, we have diffusion, facilitated transport, and active transport. Diffusion is simply moving from a high concentration to a low concentration, and some things can go directly through the lipid bilayer without needing any proteins to help them. Things such as oxygen and CO2 are small, nonpolar molecules that can move on their own through the lipid bilayer, so diffusion will work for oxygen and CO2. Facilitated transport can use channels or can use certain transporters, and we'll look at that more in a little bit. And this means that you have facilitation by a protein to move something that could not go through the plasma membrane on its own. Last, there is active transport. Active transport requires adenosine triphosphate, aka ATP. So active transport is active because it requires energy. So let's look at these in a little more detail. Diffusion is slow, and it moves down a gradient. Here we have water on two sides of a U-tube. No, not that U-tube. It has a selectively permeable membrane in the middle that is permeable to water, but not glucose. Now, we're going to do an activity in class showing how this works, but suffice it to say those glucose molecules interact with the water in such a way that there is essentially more water on the pure water side of the U-tube than on the glucose side of the U-tube. No, not that U-tube. We've done this. Anyway, the water will move from the side that there is more water to the side where there is less water, creating a pressure differential known as osmotic pressure. Why osmotic? Because osmosis is simply the diffusion of water across the cell membrane. Now, when we look at diffusion of water, specifically osmosis, we see three terms that become very handy to know. The first is a hypotonic solution. That means the concentration of solutes in the solution is lower than the concentration of solutes in the cytoplasm. Cytoplasm is a solution. Now, what this means is water will diffuse from the hypotonic solution into the cell, causing the cells to swell as water goes into them. Then we have an isotonic solution. That solution has the same concentration of solutes as a cell. Think about a saline drip if you are going to be doing some IV work. That would be an isotonic solution to red blood cells. That way you're not causing red blood cells to swell or to shrink like they would in a hypertonic solution. Like if you take a potato and put it in salt water, the potato will lose water as water moves from the potato into the hypertonic salt solution outside of it, causing the cells to shrink a little. So hypotonic, isotonic, and hypertonic are three ways of comparing solutions to cells in terms of osmosis. What about moving things with proteins, aka facilitated transport? Well, we can look at two different types of facilitated transports here. It's basically a channel protein that allows things to diffuse down their concentration gradients. No energy input is needed, but allows it to get through the plasma membrane when normally it would not be able to. A carrier protein will allow a specific number of molecules and can be very specific about how many and under what conditions they are allowed in. For example, we have a channel known as aquaporins. Aquaporins allow water in and out of cells very easily. We also have something called a sodium-glucose transporter. This will allow glucose into the cell only when it comes alongside sodium. This is why something like Gatorade, which has sodium and glucose, is so effective at getting glucose into your cells, because it comes with sodium and utilizes the sodium-glucose transporter to move both sodium and glucose across the cell membrane. Wait, I thought Gatorade was for rehydrating. Well, yes. When sodium and glucose move across the cell membrane, those are solutes. And increasing the solute concentration inside the cell means that water will now move into the cell as well, say, through aquaporins. See, facilitated transport is very useful. Active transport uses ATP to move something against its concentration gradient. Those little black balls, they don't want to be outside. They want to be inside because there's a lower concentration of those black balls inside than outside. So it takes energy to yeet them out. That is active transport. A great example of active transport is the Sodium-potassium ATP-ase. This will allow 3 sodium to move out with the help of ATP, and then 2 potassium to move back in, resetting this protein to the state where it can take 3 more sodium and with the help of ATP, put them out and then allow 2 potassium back in, resetting the protein. This occurs again and again, and we are going to see this create a chemical gradient because now there's more sodium outside than inside and more potassium inside than outside. But this also creates an electrical gradient because 3 positive charges leave and 2 positive charges enter. So the combination of an electrical and chemical gradient we are going to see as essential when we're covering neurons later on. Next, we have for large molecules, endocytosis and exocytosis. Endo, or into, cytosis brings molecules into a vesicle, cover that a little more later, from outside the cell. Vesicles are specialized organelles that transport things around in a cell. It is essentially the way of cells eating their external environment. Exocytosis brings molecules from inside to outside, such as secreting the proteins that make up mucus into your nose. Penocytosis is a specialized type of endocytosis that specifically takes up fluids from the surrounding environment. So, we've covered water, we've covered small molecules, we've covered small molecules that require transporters, we've covered small molecules that require transporters to move against their gradients, and we have now covered large molecules. What about information? Receptors will allow information to cross a membrane. A signaling molecule will bind to a receptor and cause a response in the receptor. Now, this can be something like cell-to-cell contact. Cells that are in contact with each other are giving each other signals, saying, hey, I'm here. Oh, you're here? That changes what I'm going to do on the inside. We also have hormones, which we'll cover later on. These are going to need receptors for cells to be able to know that a hormone is present. That is how information can be taken up by a cell, through the cell membrane.